Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Dr. Amit Jhala and Dr. Ethann Barnes about pollen-mediated gene flow in popcorn. Popcorn is a healthy and popular snack all over the states. Genetic limitations have long protected popcorn from harmful cross-pollination from field corn. However, recent crossbreeding efforts with Mexican corn varieties has introduced greater risk for cross-pollination between field and popcorn varieties. Amit and Ethann's work hope to assess the different factors which can exacerbate this problem. We'll talk more about this soon, but first, we wanted to thank our sponsor, Gasmet Technologies, the maker of the GT5000 Terra, the smallest portable FTIR multi-gas analyzer for greenhouse gas and environmental research. Measure carbon dioxide, carbon monoxide, methane, nitrous oxide, nitric oxide, nitrogen dioxide, ammonia, and water vapor in real time simultaneously from static or automated chambers. Visit www.gasmet.com, that's Gasmet spelled G-A-S-M-E-T, or email sales@gasmet.com for more information. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Ethann Barnes and Amit Jhala with us. Welcome to the show. Ethann grew up near Brady, Nebraska on a cow and calf ranch. He received his degrees in weed science from the University of Nebraska-Lincoln, where he researched crop and weed interactions, improving herbicide programs in soybean and popcorn, and understanding the unique set of challenges popcorn growers face in a landscape dominated by genetically modified field corn and soybean production. He is now living in North Carolina, where he is a scientist in field development for Greenlight Biosciences. Amit completed his PhD from University of Alberta, Canada, and then he was a postdoctoral scientist at the University of California, Davis, and then at the University of Florida. He's an associate professor and extension weed management specialist at the University of Nebraska-Lincoln, where he's been since 2012. He provides research and extension network for weed control in corn, soybean, sorghum, and popcorn in Nebraska. His research program is focused on weed biology, pollen-mediated gene flow, and management of herbicide-resistant weeds. For more information about him, go to agronomy.unl.edu/Jhala, that's Jhala, and we will also have a link to that in the show notes. Welcome to the show, you guys. How are you doing today?
Doing very well and glad to be on the show.
Excellent. We are glad to have you. Today we are talking about one of my favorite snacks, which is popcorn. So I'm going to throw a slight curveball at you guys and ask a bonus question before we launch in, which is, do you have a preferred way to eat popcorn, whether that's trail mix, sweet and salty, buttery? What's your preference?
I'm an air pop guy, personally, so that's definitely my way of eating it.
And I prefer popcorn with butter and little bit salt.
Nice. That is definitely the way to go. I'm more on the butter than the sweet side myself. But I'll eat almost anything. But anyway, today we are talking about popcorn, some gene flow between different kinds of popcorn. I know we had a really nice analogy to talk about that. So can you guys explain kind of some of the key terms and concepts that we'll be talking about today?
See, the complex part of this work is understanding the two versions of our gene of interest known as alleles and what they mean. So we can use an example of keys and say that each allele is a different key. If we say that the act of pollination is a doorknob, then the popcorn needs a specific key to unlock it. And the popcorn pollen has a key that matches. So the pollen is allowed to grow down the corn silk. So typically, US field corn has a differently shaped key in its pollen. So this shape does not match the doorknob of popcorn. And so the pollen cannot grow down the silk. The problem comes with the corn being introduced from Mexican corn populations in our country. This field corn's key does not matches the popcorn either, but instead it is like a master key. So despite not being the exact key needed to unlock the door, it is able to open it anyway. Thus, the field corn hybrids introduced here in our country that have the allele from Mexico are able to pollinate the popcorn, which is a problem, and that was the overall objective to conduct this research is to conduct a risk assessment of pollen-mediated gene flow from field corn, which is also known as dent corn, to popcorn.
Okay, so basically these different pollination keys are allowing kind of these hybrid corns to be formed. Is that correct?
Yes, and before we start much detail about this project and we go much in technical side, I would like to mention like we grow different type of corn in the United States. Number one is field corn, which is also known as dent corn, and which is the majority of corn we grow. Like about 98% of corn we grow in our country is field corn. And field corn is grown for different purposes. For example, it's grown for ethanol, animal feed, human food, beverages, and also field corn oil has number of industrial applications. So that is number one. And then the second one is sweet corn, the corn that we eat actually available in the grocery store and we bring it home and we eat. That is known as sweet corn. And the third one is popcorn. So popcorn is everybody familiar with popcorn, and in fact in the United States popcorn is a very healthy snack, and this is because popcorn contains high fiber, no cholesterol, and low fat, and several vitamins and minerals, and also polyphenols. So it is a very healthy snack that we prefer to eat whether we are in a movie theater or we are at home. We love popcorn. And the last one is ornamental corn, which is just for ornamental purposes. So as I told you, majority of corn we grow is field corn, about 98%, and it accounts for about 80 million acres annually in the USA. And popcorn is grown on about 2.5 million acres across the country. And it is very important crop in the state of Nebraska because Nebraska is number one popcorn producing state. We grow about 50 to 70,000 acres every year. And we are number one state producing about 45% of total popcorn produced in the USA. And so that's why this project was extremely important for our popcorn industry to conduct a risk assessment to determine the pollen-mediated gene flow from field corn to popcorn.
And why is it so important that these genes are not crossing over from one to the other? Like, what are some of the risks involved with that crossover?
So popcorn and field corn are the same species of the plant, but they bred for different purposes. So popcorn has specific traits that control the starch and seed coat of its kernels and allow it to pop. And popcorn can normally be grown side by side field corn without any pollen from field corn pollinating the popcorn because of specific properties in the pollen. And this is helpful because field corn, as I told you, is about 98% of the corn we grow in the USA. And if you go in every state, you will find field corn being raised. So it is hard to grow popcorn if we do not have a genetic barrier which can prevent the cross-pollination from field corn to popcorn. And so they look for diversity in other populations to gain helpful traits. However, during this, a trait was unknowingly brought into the US field corn population, and it can pollinate popcorn. So as I told you, in general, field corn do not cross-pollinate with popcorn because of the genetic barrier that we do have with GA1 system. But because of this specific hybrid that was bred with our US field corn hybrids, it can pollinate popcorn. And so popcorn breeders are careful to select traits that benefit popcorn, and inadvertent field corn pollination would be a problem for breeders. And so that's why, this project we conducted was kind of important, just to see how much it can outcross with popcorn.
So, you were kind of on the hunt to see how much of a risk this is and do some research into that. Can we talk about the experimental design for this project, how you were testing for these things, some of the different factors you were looking for specifically, and how you were measuring them?
Sure, yeah. So if we think about the experimental design, it was out in the open field. The first thing that we took into account was there any other corn types planted around? So we needed it to be isolated so we didn't have other research projects or other production fields accidentally pollinating our field and giving us a false positive. So we started with planting the field corn that had this trait that can allow it to pollinate popcorn in a very small square in the middle of our field. So we planted about 4,000 square feet. So really small square of this field corn. And then around it, we planted white popcorn. So the kernel color of this popcorn is white, and we planted about 3.5 acres in the square around this field corn. So some of the things that we were looking to measure were all during pollination time. So for measuring cross-pollination, the traits that we're looking for that could affect cross-pollination are things like wind speed, distance, humidity, direction, as well as other wind factors like wind gust or the period of calm wind in our experiment. Ultimately, at harvest, we looked at kernel color. So because the field corn in the middle was a yellow kernel color and the popcorn around the outside was white, we can actually measure gene flow by looking at the kernels of the popcorn. So yellow is a dominant trait when we consider kernel color. So all we did was harvest the ears and then count the number of yellow kernels on white popcorn cobs. So that gave us the amount of gene flow from field corn to popcorn.
Okay, I have a few questions off of this, and I hope you'll forgive my ignorance of kind of the corn life cycle here. So first question, are like pollinators an issue for corn pollination like bees and butterflies are for flowers? I mean, I guess I've never seen bees around cornfields. Is that a thing? Like, do pollinators pollinate corn?
Yeah, so that's a great question. Really, it's a minimal amount of pollination that could be affected by pollinators, but it is definitely something to consider. So insects, not just pollinator insects, can ultimately carry pollen on them from one corn plant to another. So it is a factor. It's definitely a small factor with corn pollination.
Okay, that's good to know. And then my second question relates to these corn kernels. So, obviously with eye color, it's not like you usually end up with like, oh, I have a part, like a 1/4 blue eye and 3/4 brown. How does that work with corn? Is it like if it was more pollinated by this other corn, you would see more kernels? Because you mentioned counting them specifically. So how does that kind of ratio work out within a single cob of corn?
Right. So each kernel has a silk attached to it, and that's how it's pollinated. So a single pollen lands on each silk and pollinates that particular kernel. So the cob could be anywhere from one yellow kernel to 100% yellow kernel. It's all about chance. So yeah, you can really track the amount of gene flow on a single cob. Of course, we looked at a lot of cobs, a lot of ears of corn to determine the rate of gene flow. But if we think about how the genetics is transferred in the pollen, if a yellow field corn pollen pollinates the white popcorn, it's going to always be yellow. So there is no ratio right away with the kernel color. if you were to grow those yellow kernels, some of those plants would produce pollen that would turn out to be yellow corn, and some would produce pollen that turns out to be white. So the inheritance of that gene is really displayed when you plant those kernels.
Okay. That makes a lot of sense. I did not know that was how corn pollination works, so I'm already learning tons here. And then a more specific question for the experimental design is, I'm assuming you didn't check like every cob within all of this land. So how did you decide like what areas you were going to pull corn from for your counting?
Yeah, so if we think about how it was laid out, we had a small square in the middle, which is the field corn, and the majority of it around the outside was a square of white popcorn, right? So we just sampled along transects, starting at in each direction. So we had north, east, south, and west, and then also northeast, southeast, southwest, northwest. So we had 8 different directions, and from the center, you can think of it like a wheel with spokes. So those directions went out, or those 8 directions went out all the way to the edge of the square, which on the northeast southwest line was 50 meters from the center block, and in the corners went out to 70 meters. So we then just sampled at various lengths along that transect. So we did a lot of close distances because we know pollination, the highest rates of pollination, that is, would happen at the closer distances more. And then we also sampled out to 30, 40, 50, 70 meters in the corners. Okay.
So then my final question off of that is, obviously you're testing for a lot of things, that you mentioned, the wind speed or direction, proximity, humidity, things like that. How do you know like which of those factors is causing the pollination at any given time? Is it just something where you are, monitoring throughout the season? How do you know that it wasn't, because of this one humid day versus it was because of this windy day, or how do you split those out?
Right. So that's a great question. So ultimately, we're collecting that data throughout pollination from a weather station that's sitting right in the middle of this experiment. So the wind meter on this weather station was collecting wind speed from just above the popcorn tassel. So we're getting real-time weather data, and that includes, humidity, it includes wind speed, the gust occurring, and then we're figuring out how that is correlated to the amount of gene flow at those different distances and those different directions by putting it into several different models. And so we're looking at those factors alone. Is one of those factors, let's just pick one, say wind gust, is that one factor enough to predict the ultimate amount of gene flow at those different distances? Or is it a combination? So we looked at those individually. We looked at each combination, so just 2 combinations, 2 different factors together, all the way out to all of those factors in a different model. So that ultimately tells us the importance of those factors. We can compare those models and it'll tell us the best fit. So at the end of the day, the best fit model has the parameters in it that most impact gene flow.
Okay. Hi, everyone. I hope you're enjoying the show. Interested in learning more? Amit and Ethann's article, Risk Assessment of Pollen-Mediated Gene Flow from GA1M Field Corn to Dense Sterile GA1S Popcorn, published in Crop Science, will be freely available for the next 2 weeks. You can find a link to it in our show notes. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Thanks again also to our sponsor, Gasmet Technologies. Conduct greenhouse gas flux measurements in the field using the GT5000 Terra. Weighing 20.7 pounds, splash-proof IP54 rated, internal pump and battery, and vibration resistant, the GT5000 Terra is a robust and portable multi-gas analyzer for fieldwork. Visit www.gasmet.com or email sales@gasmet.com for more information. Thank you for being our sponsor. Let's get back to the show. Well, that seems like a great time then to transition. What did you find?
Yeah, so the biggest factor in affecting gene flow in this experiment was actually distance. This isn't really that surprising, but if you think about the collection right across from the field corn, we had relatively high gene flow, so nearly 5% gene flow. So that means that the popcorn in the row next to the field corn had the highest amount of gene flow. And then as we went all the way up to 50 or 70 meters away from the field corn, it declined drastically. That being said, we still had detectable gene flow all the way up to 50 and 70 meters. This isn't surprising for a couple of reasons. The biggest reason is that it's field corn popcorn, and it's been studied a lot. So if we think about the actual size of the field corn pollen, it's actually quite large. So you wouldn't expect, compared to other angiosperms, you wouldn't expect corn pollen to travel very far. It's heavy, right? So you're going to need either a lot of wind or some other, say, an insect to carry that pollen, several miles. When we're thinking about a relatively small area, like up to 70 meters, we can still see it naturally moving that far, but at a very low rate. So if we think about the other factors we tested, it's not to say that those aren't important. They're just not major players in detecting the amount of gene flow and the distance of gene flow. So wind speed, of course, is very important for corn pollination. You have to have some wind movement, some air movement to get pollen shed to happen in the first place. But ultimately, it wasn't a major factor in the rate of gene flow for us.
Okay. And so you said that it was about 5% was your highest. I think my response is like, oh, wow, like that's not actually that much. So the concern is basically, even though 5% doesn't seem like a lot, the concern is that, now if you use the kernels from that corn for future plants, then those will have those genes and then potentially keep spreading them, like in their offspring, right?
Yeah, so that is the major concern. It's important to point out with this research, we're not really measuring, we're not looking for the final rate of gene flow. So even though it's 5%, that's really a factor of our experimental design. We're more looking at the factors that affect that gene flow. So it's only 5% in part because we're testing mostly popcorn in this field and a very small amount of field corn. So if our center block was the same size as our white popcorn border, then we would expect more gene flow, right? So if we planted 3.5 acres of field corn next to 3.5 acres of popcorn, maybe those border rows would have up to 50 or 40% gene flow. But we had much more popcorn pollen in that field just because we planted more popcorn plants. So we only saw 5% gene flow. But to get back to your question, yes, that is the major concern. That's the biggest concern, really, is if it's in a breeding program and it's not white popcorn, right? Maybe he's growing yellow popcorn next to yellow field corn, and he doesn't know that his field corn could pollinate that popcorn. That breeder could then select those seeds to move on in his breeding program without realizing that there's a major concern. It's half field corn. And so that's really the major concern, is when it's not so obvious, when it's the same kernel color and those kernels or those seeds are moved forward in a breeding program.
Okay.
And just to add to that point, and that's true, Abby. In general, you feel like, it's only 5%, but 5% for scientists or for this discipline, like if I am a popcorn breeder And if I have a 5% outcrossing with any other type of field corn or popcorn, which is a major concern, and that is something it can reduce the overall quality of popcorn in generation, and it reduces the overall quality of popcorn.
Yeah, absolutely. Little goes a long way in these for sure. Yeah, so another thing that I know you mentioned in your paper that affected this experiment a little bit is that the pollination times for these two varieties didn't exactly sync up. Can you talk about that and how that's a factor as well?
Yeah, so all different corn, both popcorn and field corn, have different times to maturity. They have different times to pollen shed. So in the first year of this experiment in 2014, we planted the popcorn and the field corn at the same time. So the field corn actually had a longer day to maturity, and it started to pollinate after the popcorn. They did sync, so we did have open silks on the popcorn at the same time as the field corn was shedding pollen, but it was only a few days overlap. So in the second year of the study, we actually offset the planting. We planted the field corn first, and then a week later came in and planted the popcorn. So then when they pollinated, they synced at the same time. There was open silks the whole time that the field corn was pollinating. So that's a huge factor that can influence gene flow, right? And especially from corn to corn. And that's one of the ways that people can prevent cross-pollination in cornfields, is just offsetting the planting enough that you don't have viable pollen from the field corn when the popcorn has silks receiving pollen.
Sure. Yeah, that's a great strategy for sure. And so now that we've kind of established that this is definitely a potential threat to this crop and breeding programs, what is some future research questions that we still have to answer moving forward?
So in popcorn now, we have conducted this study, and now maybe if in future we have a chance, we want to see how much is the gene flow at a landscape level. Like our study was conducted, our overall distance was maximum 200 feet from the center field corn we planted, and we planted popcorn up to 200 feet distance in all the directions. And now the next step we want to see at a landscape level. Like, so for example, if we have a field corn field of about couple of acres, and then if we have a popcorn growing in about 10 to 20 acres, and then what will be the chances of outcrossing and up to how far the pollination can occur, and that can help us to maintain certain level of isolation distance, that we can say like, okay, if we can maintain this much isolation distance, we will not see any outcrossing event. So that will be the next step in this project.
Great. Yeah, that is wonderful. Gotta keep those crops protected. Yeah, I really enjoy this project. It's a lot of fun. So if people want to learn more about these issues, whether that's the crossbreeding or popcorn crops in general, where can they go for more information?
So studying pollen-mediated gene flow is a primary objective in my lab at the University of Nebraska-Lincoln. So this project was specifically to study gene flow from field corn to popcorn, but the same concept we have used in number of other projects. For example, we are conducting studies to evaluate the pollen-mediated gene flow from herbicide-resistant to susceptible weed species. Means we have at least 6 weeds in Nebraska have evolved resistance to glyphosate, which is a chemical being used by growers for weed control in glyphosate-resistant corn and soybean fields. And those are 2 major crops we grow in the state of Nebraska and several other states in the Midwestern United States. So we have conducted number of studies to evaluate how the distance and other weather parameters can play an important role to disseminate the resistance that we do have. So now, after conducting all these studies, now we can say like pollen-mediated gene flow is also playing a very important role to disseminate the resistance evolution that we do have. So once weed can evolve resistance, it can also cross-pollinate with other susceptible weed species, and that is how this resistance is actually spreading across the field and then across the state. Especially, it also depends on reproductive biology of the weed species. For example, there are certain weed species which are dioecious species, means male and female plants are separate, and that means outcrossing and gene flow is the only way for them to survive. And so for them, the chances of outcrossing is more if you compare with some other species where they are monoecious species, means male and female reproductive parts are on the same plant, that reduces the chances of outcrossing. And same concept we can discuss for corn. So for example, corn is an open pollinated species. That means it increases the chances of pollen mediated gene flow if you compare with other crops. For example, soybean or wheat, they are extremely self-pollinated species, and chances of outcrossing will be very, very minimum. So we have all this type of projects going on in the lab, and all this information is available on our lab webpage, agronomy.unl.edu/Jhala, Jhala. So on this website, we have a number of projects, and PDF of our published papers are available on number of projects we already have accomplished and what we have new in the lab.
Great. We will definitely include a link to your lab on our show notes so listeners can check that out. Sounds like a ton of awesome work you're doing. So thank you for doing that. And if people then want to take the next step and get involved in this, if they're interested in these fields, where can they go? What can they do to get involved?
They can contact us and we can provide more information on each study that we have conducted. Additionally, we also write a number of extension articles on our University of Nebraska-Lincoln extension portal, which is cropwatch.unl.edu, which provides all this science-based information in very easy to understand language. So we also conduct a number of extension field days where growers can attend, and face-to-face they can see the results of our studies and they can learn more about our projects and overall weed management and gene flow from crops to weeds, and also from weeds to their closely related species.
Great. Yeah, we will include links to that as well. So check it out, get involved. I have one final question for you guys today. Thank you for all of your wonderful answers so far, which is, what is one fun fact that listeners would not know about you if all they had was your research?
For me, I like to make vodka-based cocktails to my friends and students. Every year I invite my students at home and I like to make some very nice quality cocktails and they enjoyed it. So this is something most people don't know about me. I can be a very good bar attendant.
Ooh, is there a particular recipe or flavor that you enjoy the most?
Yeah, generally strawberry-based vodka with some good cocktail mixtures is my favorite, and my friends like it.
Yeah, I can confirm that. It's definitely something to look forward to every year. So,
all right. Well, wonderful. How fun. Ethan, what is a fun fact about you?
Yeah, so I spend most of my free time outdoors. I have 2 Shetland sheepdogs. I like to take hiking, going for walks. We're actually pretty lucky that they didn't break into the room here and include themselves in the podcast, I think. So,
I mean, I would have loved to see them, have seen them, so I wouldn't have minded. But seeing as our listeners wouldn't have gotten to enjoy that, I guess that's fair that they didn't join in. Well, wonderful. This has been a great interview. Thank you for being on the show and for all of your hard work to make sure that we can still enjoy popcorn for years to come.
Thank you for having us, Abby.
Thank you for listening to Field Lab Earth. You'll find a link to today's paper and other resources for this episode in our show notes or on our website. If you have any questions, comments, or recommendations for show topics, please contact us at podcast@sciencesocieties.org or on Twitter @FieldLabEarth. If you'd like to hear more content like this, please subscribe and don't forget to rate and review us on Apple Podcasts, Podchaser, Stitcher, or anywhere else you find your podcast if you like our show. We are also available on Lyceum, the world's first audio learning community, where you can join our discussion group and comment on each episode. This podcast is a joint production of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. Special thanks to Lobo Loco for the use of their song Spook Castle on the intro and outro of our show. Opinions and conclusions expressed by authors are their own and are not considered as those of the American Society of Agronomy, Crop Science Society of America, Soil Science Society of America, its staff, its members, or its advertisers.